Hello students, welcome to EPG Pathshala. I am Dr. Nidhi Garg from Deshbandhu College, University of Delhi. Today we are going to talk on module Microarray Part 1 from the paper Molecular Genetics. So students, let us see what we are going to learn in this module. You will be able to know how microarray experiment is performed, learn about different types of DNA microarrays, evaluate the importance of microarray in detecting gene expression, know the current and potential applications of DNA microarray. A microarray consists of an array which is an orderly arrangement of samples such as DNA, RNA, proteins, etc., immobilized on a miniaturized solid support. The sizes of the sample spot are usually less than 200 microns in diameter. Therefore, a single microarray contains thousands of spots. These thousands of spotted samples are known as probes, and the identity of each spot is known. The solid support used are either microscopic glass slides or they are silicon chips, and in some cases the nylon membranes are also used. T. Wen Chang in 1983 introduced both the concept and the methodology of microarrays in the form of antibody microarrays. Although the antibody microarray was the first to be developed, but the most popular type is the DNA microarray, also called as the gene chip. A microarray is used for assessing large amounts of biological material, which may be DNA, RNA, oligosaccharides, or proteins, using high throughput screening with parallel processing and detection methods. Types of microarrays. There are several different types of microarrays: DNA microarrays, protein microarrays, peptide microarrays, tissue microarrays, cellular microarrays, chemical compound microarrays, antibody microarrays, carbohydrate arrays, phenotype arrays. DNA microarrays are also called as DNA chips or biochips. They consist of an orderly arrangement of samples or spots which can be either DNA, cDNA, or oligonucleotides attached to a solid surface. The principle of DNA microarray is based on the ability of single-stranded DNA, cDNA, or RNA to hybridize to the complementary strands of DNA, cDNA, or RNA in solution with high specificity based on base pairing rules. Each DNA spot comprises of only picomoles of specific DNA sequence, which are known as probes. Probes are short section of gene or other DNA element which is used to hybridize a cDNA or cRNA sample known as the target. DNA microarray technology has evolved from the technique of Southern blotting. Using DNA microarray, one can quickly and efficiently analyze the expression of several genes in a single reaction. Thus, it provides information regarding gene expression and gene discovery. The gene arrays were initially made by spotting cDNAs onto filter paper using a pin spotting device. The miniaturized microarrays were used for the first time in 1995 for gene expression profiling. But it was in 1997 that the complete genome of Saccharomyces cerevisiae was spotted on a microarray. The microarrays can be constructed by using two methods. The first is direct synthesis of oligonucleotide probes on solid surfaces, while the second method is that the probes are first synthesized and then attached to a solid surface by covalent bond via surface engineering to a chemical matrix, which could be epoxysilane, lysine, aminosilane, polyacrylamide, etc. The orderly arrangement of probes is important because the location of each spot on the support is used for the identification of a gene. The hybridization of the probe and target is detected using either a fluorophore, silver, or chemiluminescence labeled targets. It is also used to quantify the relative abundance of nucleic acid sequences in the target. The solid surface used for constructing the microarray can be glass, or a silicon chip. Types of DNA microarrays. There are basically two different types of DNA microarray. One is the glass slide microarrays and second is the Epimetrics gene chips. Types of DNA microarrays. Glass slide microarray. The probe DNA is attached directly on the surface of a glass microscope slide. In glass microarray, two samples can be hybridized on a single chip for the direct comparison of the samples. Longer probe DNA or cDNA sequences at times cross-hybridize with the other genes due to sequence similarity. Probe DNA can vary in size from a medium-length oligonucleotide, say 60 nucleotides, to an entire cDNA clone or an even larger DNA fragment. The DNA is deposited on glass slide in two ways, either using printing or spotting. It is done using an inkjet printer. This technique was developed for depositing ink droplets on paper. It consists of 4 cartridges for depositing the bases adenine, thymine, guanine, and cytosine on array substrate at defined positions. The gene sequences to be synthesized are taken as a text file and given as input to the synthesizer. The information contained in the text file is used by the base delivery system, which subsequently places the probe on the defined position in the array. Spotting. This is done using robotic spotters that use a set of fine-tipped metal pins that act by capillary action. Many pins are mounted together and simultaneously dipped into different wells of a microtiter plate containing different types of DNA samples. The DNA is amplified from cDNA or genomic DNA through PCR. Spotted DNA microarrays. They were used by research scientists world over and were produced from their own labs. They were easily customized for each experiment as the researchers were able to choose both the probes and the printing locations on the arrays. Also, the probes were synthesized in their own lab and then spotted on to the arrays. The researchers were able to prepare their own labeled samples, which were hybridized to the array, which was scanned with their own equipment. This technique provided a comparatively low-cost microarray which could be customized for each study. Spotted DNA microarray. It has also led to cost cutting as the purchase of expensive commercial arrays containing large number of genes which were not of interest was avoided. Several scientific publications have indicated that the in-house spotted microarrays do not provide the same level of sensitivity as the commercial oligonucleotide arrays do. This might be due to the small batch size and reduced printing efficiencies. Epimetrics gene chips. It contains about 10 to 20 nucleotides for each gene that are non-overlapping and 20 to 25 bases long. These oligonucleotides are selected using softwares that scans the whole genome to select regions that are unique in the genome to prevent cross-hybridization. The oligonucleotides fit into the empirically derived design rules that decide the ratio of GC versus AT content and reduces the possibility of self-hybridization of oligonucleotides creating hairpin structures. The oligonucleotides are synthesized directly on a solid support by photolithography, a technique that is used for printing the computer circuits. This technique was pioneered by Epimetrics, hence the name was kept as Epimetrics gene chips. Each probe on the chip has a corresponding mismatch probe which has a single base difference at the 13th nucleotide. Fluorescently labeled RNA is hybridized to the array. The abundance of each RNA is calculated on the basis of the amount of hybridization seen for the entire probe set. Correction is done for hybridization to the mismatch probes that indicates the possible non-specific effects. Only one sample can be hybridized per chip, hence, direct comparison of two samples cannot be done, something which we see in case of glass chip microarrays. Limited amount of probe sequence helpful in limiting cross-hybridization but can be problematic because it might miss alternative splice sites, can show bias towards one end of the transcript, or we can say that it is heavily dependent on good genome annotation, which might not always be possible. Differences between spotted microarray and Epimetrics gene chips. If we consider the features like the technique of manufacturing, we see that in case of spotted microarrays, the synthesized probes are fixed to the slide, whereas in case of gene chips, the probes are synthesized directly on the slide. In terms of hybridization method, the two labeled samples are hybridized to the same microarray for comparison, called competitive hybridization in case of spotted microarrays, whereas only one labeled sample can be hybridized to each chip in case of the gene chips. In terms of analysis, the spotted microarrays allows the measurement of relative differences between the signal intensity of two targets binding to the same spot of DNA, whereas comparisons are made by measuring the hybridization intensities on two different chips in case of the gene chips. But if we talk about the shortcomings of the two techniques, in spotted microarrays, the variability in the quality of the spotted DNA, both in terms of amount and in the integrity. The competitive hybridization measurement compensates for the differences in the quality of the spot. The gene chips made with photolithography have higher reproducibility from slide to slide, which makes the technique of competitive hybridization less important. Basic steps to DNA microarray. Either individual cells or the desired tissue containing the genes of interest are required for analysis. The mRNA is isolated using the extraction buffer. The mRNA at its 3' end has a string of adenines, which is used as a primer, and the mRNA is converted into cDNA. The cDNA molecules are used as targets, which are labeled with fluorophores to help in their detection. The cDNA is incubated with the chip DNA. Some of the cDNA will hybridize with the probe molecules due to complementary base pairing. Washing is done to remove the unbound cDNA and to know what has actually bound to the DNA chip. The microarray is exposed to laser light of specific wavelength to excite the fluorophore molecules, which emit light of a particular color. Finally, the results are interpreted using the desired software. Let us take up an example to study differential gene expression. Now, in normal cells and tumor cells, certain genes are expressed while the expression of the other genes is masked. Thus, in both the cases, differential gene expression can be detected and studied using the technique of microarray. For this, first of all, we extract the mRNA from both the normal and the cancerous cells. Secondly, the mRNA is converted into fluorescently labeled complementary DNA, also known as cDNA. The cDNA of normal cells and tumor cells are differentially colored. The different cDNAs are mixed and then added to the cDNA spotted onto the microarray for incubation. Washing is done to remove the unhybridized cDNA. The microarray is read with the laser and the amount and pattern of fluorescence is recorded and the data is analyzed and interpreted using the appropriate softwares. Reading a microarray. The Microarray chip or the DNA chip is placed inside a dark box and then scanned using laser, which emits light of a particular wavelength which detects the bound fluorescent labels. Separate laser is used for each color, such as one for green, one for red. Both the information and images are sent to the computer for analysis. Image analysis involves, first, suppression of noise, second, the detection and localization of the spot, which also includes extraction of the background intensity and identification of the spot position, boundary and size. Third, quantification of data and quality assessment. Analyzing the microarray data. When the fluorescently labeled cDNA hybridizes into the microarray and the detection is done using the laser light, a ratio image is created by the computer. For example, the green images signal expression under one condition, the red images signal expression under one condition, the yellow images signal expression under both conditions. As the microarrays measure the expression of thousands of genes simultaneously, large amounts of data is generated, which requires computers both for the storage and its analysis. Types of data analysis. There are three different types of microarray data analysis. The first is gene by gene analysis. Here, the data is analyzed by looking at the expression levels of individual genes. This requires the use of statistical techniques which are also used in several other biological experiments. The second is categorizing groups of genes. Here, for examining microarray data as groups of genes rather than single genes, the researchers use clustering algorithms as it offers a higher level of analysis. The third is deducing patterns of gene regulation. For interpreting the microarray data to know the patterns of gene regulation, the analytical methods are still under development. Let us talk about the applications of DNA microarray. The first being expression analysis. Microarray technique is extremely useful in identifying the expression levels or quantity of genes, both mutant and normal. It also helps in making comparisons between tissues and genotypes. The second important application is in mutation analysis. For the detection of mutations, the researchers use genomic DNA as a probe. At times, the genes differ from each other in just a single nucleotide base. The difference of a single base between two DNA sequences is known as single nucleotide polymorphism. DNA microarray is useful in detecting SNPs, multiple alleles of a gene, and mutated genes responsible for a particular disease. Applications of DNA microarray. Comparative genomic hybridization. It is useful for the assessment of genome content in either different cells or in closely related organisms. This provides knowledge of the presence or absence of certain genes in closely related organisms. For example, genes involved in a particular metabolic pathway. Gene ID. Small microarrays are used for checking the presence or the so-called IDs of particular organisms that may contaminate the food and feed, mycoplasmas that may infect a cell culture, or pathogens causing particular disease. This is done by combining PCR and microarray technology. Toxicological research. Microarray technology has been useful in establishing the impact of different types of toxins on the cells. Toxicogenomics has helped in establishing a correlation between response of a cell to toxicants and the subsequent changes in a cell's genetic profile upon exposure to such toxicants. Gene discovery. DNA microarray technology has played an important role in the identification of new genes and has helped in gaining knowledge regarding their functioning and expression levels under different conditions. Drug discovery. Microarray technology has proved to be very crucial in the development of pharmacogenomics. Pharmacogenomics is the study of correlation between the genetic profile of patients and their response to drugs. Thus, by studying gene expression in both a diseased and a normal cell, the proteins synthesized by the diseased genes can be easily identified. This information can then be used by researchers in synthesizing drugs that can combat with these proteins, thereby reducing their effect. Disease diagnosis. DNA microarray technology is helpful in learning more about diseases like heart disease, mental illness, infectious disease, and cancer. Before the evolution of microarray technology, different types of cancer were classified on the basis of tumor development in particular organs. But now, the researchers can classify cancer based on patterns of gene activity in the tumor cells. This will allow the pharmaceutical industry to develop drugs that directly to the specific type of cancer. Tiled microarrays. Tiled microarrays contain either a particular region of a genome or a complete genome at high coverage. The probes are designed such that they cover almost every base pair of the genome except the simple sequence repeats. There is absolutely no bias towards known transcribed regions. These microarrays can only be used for those organisms whose genome has been completely sequenced, which aids in their identification. Protein binding microarrays. These microarrays are used for identifying the transcription factor binding sequences, also known as motifs. Instead of single-stranded DNA probes, the array contains double-stranded DNA probes to which purified protein is added for hybridization. The protein is detected using fluorescently labeled antibodies specific to the protein. This technique involves the use of either real genomic sequences or designed oligonucleotides. Validation of microarray data. There's no way by which one can validate the complete microarray data. Thus, it is very important to verify any key findings using techniques such as northern plots and quantitative RT-PCR. In fact, real-time quantitative PCR is usually the most common method of choice. Important concepts in microarray experiments. The first being experimental design. While designing a microarray experiment, the below mentioned points must be considered. First, technical versus biological replicates. The technical replicates refer to the repeat hybridizations carried out using the same RNA isolate, whereas the biological replicates refer to the use of RNA that is isolated from separate experiments or experimental organisms. Thus, the technical replicates reduce variations that arise due to hybridization or imaging, while the biological replicates are indispensable to demonstrate a properly controlled experiment. Second is amplification of RNA. If a researcher is working with very small number of cells, then usually linear amplification methods are used for increasing the amount of RNA. But as of now, it is unclear to what degree this might affect the results, particularly with respect to rare transcripts. The third is dye swaps. If two color arrays are used, then one must hybridize the replicates using a dye swap strategy. This involves the reversal of the colors or the labels between the two replicates. This is done to rule out the possibility of biases in hybridization intensity due to the use of dye even when the sequences are the same. Fourth being reference samples. A reference sample must be used in one channel on each array as it will hybridize to most of the features. By using a reference sample, one can compare between different experimental conditions as each of the experimental condition is compared to the common reference. The experimental design for microarray must always be discussed with a statistician before starting because of the expensive nature of the experiment. Statistical processing and analysis. This step involves the use of several statistical softwares to analyze the data obtained for interpreting the results so one can reach to a logical conclusion. So students, let us now summarize what we have learned in this module. Firstly, a microarray consists of an orderly arrangement of samples such as DNA, RNA, proteins, etc., immobilized on a miniaturized solid support. These thousands of spotted samples are known as probes, and the identity of each spot is known. The solid support most commonly used are either microscope glass slides or they are silicon chips. A microarray is used for assaying large amounts of biological material, which may be DNA, RNA, oligosaccharides, or proteins in the form of antibodies, peptides, receptors, etc., using high throughput screening with parallel processing and detection methods. DNA Microarray is a high throughput technology that comprises of an array containing a series of thousands of microscopic spots, each containing picomoles of known DNA sequence. The probe in DNA microarray can be a short section of a gene, a complete gene, genomic DNA, or other DNA element that is hybridized to a cDNA or genomic DNA sample called target under high stringency conditions. DNA microarray measures the changes in gene expression, involves gene discovery, pharmacogenomics, toxicological research, comparative genomic studies, or detecting changes in the nucleotide sequence which might cause a disease condition. Data produced by microarray technology must be validated using techniques such as Northern blotting and RT-PCR. Thank you.